In this video, we will discuss about the isoferulic acid impact on the in vivo studies or on the mouse which are infected with pancreatic cancers. So first what they check is that they check the tumor weight and tumor volume, and what they are able to find out is that with increase in the dosing of this isoferulic acid, they see a decrease in the tumor weight, and this decrease in the tumor weight and volume in terms of the volume is quantified in the panel B, where we can see that where isoferulic acid's concentrations is increases, there is a decrease in the tumor volume compared to the control case where tumor volume increases tremendously at 21 days. So this indicates that isoferulic acid has some anti-tumorigenic properties. And in terms of the tumor weight, the quantification is shown in the panel C, where we can clearly see that the tumor weight also goes down with dose-dependent increase in the isoferulic acid. That is, when isoferulic acid is when the isoferulic acid concentrations are higher in the mice model, we see a significant decrease in the tumor weight compared to the control case where no isoferulic interactions is there. Thereafter, they check the apoptotic factors using the tunnel assay, and in the tunnel assay also they are able to see that in the case of control, they do not see too many apoptotic cells, whereas when they use isoferulic acid, they see increase in the apoptotic cells with increase in the concentration of isoferulic acid. It can be compared from panel from figure B and C of panel D. That is, with the increase in the concentration of isoferulic acid, they see more apoptotic cells. And also, and this is the zoom in view. Bottom row of the panel D is the zoom in view, where we can see that apoptotic cells are higher when they are treated with isoferulic acid, and it even goes higher when the isoferulic concentrations are high. And in terms of the signaling pathways, that they have find out that NK kappa beta pathways goes down regulated when the isoferulic acids interacted with the cells. So whether these things are also happening in the in vivo studies or not, they check the factors for the NF kappa beta pathway, that is phosphorylated IK beta alpha, and they see that the signals for the phosphorylated IK beta alpha goes down along with that P65. So both of this also these results both of these results all of these results are in line with their observations in the in vitro studies. So from this they finally conclude that it's the isoferulic acid that is isoferulic acid impacts on the mice model in a similar way as it impacts in the in vitro lab studies, as they did they checked the isoferulic acid impact in terms of the tumor weight and tumor volume, and to show that it has anti-tumorigenic properties. And they also checked the apoptotic factors using the tunnel assay, where they find out the same trends that what they have observed in the in vitro studies, and the other things that they have find out, the signaling pathways that got dysregulated in the in vitro studies when the cells are treated with isoferulic acid, it is NF kappa beta pathway, and they are able to see the similar results here also. That is, the factors for the NF kappa beta pathway, that is phosphorylated IK beta alpha, goes down, whereas the normal IK beta alpha goes up because cell wants to prevent apoptosis, but the phosphorylation does not occur as the isoferulic acid interferes in some way to prevent the phosphorylation of IK beta alpha. And same things they have seen for P65, that is, its signals also phosphorylated P65, its signal also goes down, whereas regular P65 signals looks like it goes up or remains same. So in this case also the phosphorylation of P65 gets impacted by the isoferulic acid. So I hope now you understood how this isoferulic acid has some antitumorigenic properties that are shown here on in vivo studies, that is on the mice models. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.